as I just mentioned, the plant cell will have a cell wall and therefore cleavage is not possible. The preparation for the division in a plant cell starts in the G2 phase. There is a formation of a pre-prophase band just beneath the plasma membrane with the help of actin and some microtubules. This sets the division of plane. The plane of division. So now let's imagine that the plane of division again here is this. It goes from front to the back and in between there are chromosomes aligned. Now as the cell cycle will progress and the cell will reach the late anaphase, there will be some changes in the type of actin or microtubules, the constituents and the type. However, at the end, in the late anaphase, when the cells are moving away from each other, there will be formation of a cylindrical structure, and this structure is known as a phragmoplast. It will be present somewhere between the two cells. As the poles are decided and the nucleus starts to form during the telophase, this phragmoblast starts to extend. What happens is that the Golgi apparatus in a cell, we know that it is very useful in the packaging of materials. So now the Golgi apparatus that are present in the cytoplasm will have some vesicles that will be filled with the materials that are needed for the formation of a cell wall. These vesicles will start to fuse with this phragmoblast and the cell wall precursors will start to release, and the phragmoplast will then start to grow in both the directions. As the cell wall precursors are laid down, there will also be a presence of the microfibrils, the polysaccharides, the cellulose that is needed for the cell wall, and finally at the end, this will fuse to both the sides, and this region will then become the cell wall and the middle lamella part of the cell. As it happened in the plant cell, some of the endoplasmic reticulum may be present in the middle region, and they will form connection between the two cells, and here in plant cells, those are known as desmosomes. This is the division of a plant cell. As you can see, it is different from the animal cell in the sense that it has a cell wall, so the cleavage can't occur, and the division starts from the middle to the opposite ends, and there is a formation of a structure known as a phragmoplast. So this is how the cell cycle progresses and the cell divides into two daughter cells. However, there are some drugs that can be present that can inhibit this cell division. For now, we are going to look at just the three major ones. The first one is colchicine. This is a drug that is used very significantly when we have to observe the chromosomes. What colchicine does is that it inhibits the microtubule formation. And as you might remember, the microtubule formation is very important for mitosis, where the cells have to align in the middle with the help of their kinetochores and these microtubules. Therefore, we can say that it inhibits the metaphase of the cell cycle. Another drug that is used to inhibit the cell cycle or the cell division, to be precise, is Taxol. Now, one place you might have heard the name of Taxol is in it is used a lot as a chemo drug, that is in the cancer treatment. Taxol allows the formation of these microtubules, but it does not allow the shortening. And if you remember, according to PMAT, A was the stage, anaphase was the stage when the chromosomes or the sister chromatids had to move away from each other. Now, if the microtubules are formed but they do not shorten, the anaphase will be inhibited. And the last drug that we are going to talk about here is Cyto Calcium. Yeah. What Cyto Calcium does is that it does not allow actin to work. It inhibits the elongation and all of that actin myosin head meeting. That is the basic principle of muscle contraction. It does not allow it. And as actin is not allowed to elongate and contract, it basically inhibits the cytokinesis. As actin is a protein that is used both in the animal and the plant cell cytokinesis. With this, we mark the end of cytokinesis and in general our cell cycle. In the next video, we are going to understand the differences as well as the regulation of the cell cycle. If you like this video, don't forget to press the like button and do subscribe to my channel and press that little bell icon for getting all of the updates whenever I upload a video. Also, do tell me in the comments below what you thought of this video and if there's something that you want me to start next. Thanks for watching.